I'm going to present our lab's work on using large language models to predict antibiotic resistance in Mycobacterium tuberculosis. So if you're not familiar with what tuberculosis is, or as it's more commonly called, TB, it's an illness that affects a patient's lungs, and it's caused by the bacterium Mycobacterium tuberculosis. In the WHO 2024 TB or Global TB Health Report, they documented that there were over 10 million cases of TB worldwide, with over 1.25 million deaths. And of course, of those 10 million cases, we also had over 175,000 people treated for multi-drug resistant tuberculosis. So needless to say, TB is definitely a global health concern, especially multi-drug resistant TB. So the way that drug resistance is determined in tuberculosis is that the sample is exposed to varying concentrations of an antibiotic, and then the minimum inhibitory concentration, or MIC value, is recorded for each antibiotic. And that's pretty much the minimum amount of the antibiotic needed to inhibit the growth of that bacterium. So there have been efforts to create data sets that compile the genotypic and the phenotypic data for AMR in tuberculosis. One such data set is the Comprehensive Resistance Prediction for Tuberculosis. At the time of this work, it was the largest, or one of the largest, phenotypic and genotypic MTB data sets. We had access to 12,289 total isolates. And as you can see from the map, it's quite comprehensive. It spans quite literally places all over the globe, or it has samples from all over the globe. And the data set is composed of the AST data for 13 different antibiotic drugs from 3 different treatment lines. So first line, second line, and then you have the last resort or new or repurposed drugs. So our principal goal using this data set, we wanted to see if we could create a large language model for the prediction of antibiotic resistance just purely from the sequencing data or the DNA sequence. So this is a high-level overview of the workflow that we use to develop and evaluate our model. So it started off with us acquiring the cryptic data set. We were able to get 12,185 total samples, which was then further split into a development and an evaluation set. However, before we could use this data to train or evaluate our model, of course, it had to be pre-processed. So that's the first step here. And I'll just go through and just briefly introduce how we went about pre-processing our data for use with the large language model. So from the reads, we use spades to assemble them. And then PROCA was used to annotate 273 different MTB genes. Now, of course, some of these genes were fragmented, so we had to use BLAT to align them back to the H37RV reference genome for MTB. And then from there, we could reconstruct some of these genes that were fragmented. And at the same time, we also extracted any upstream or downstream intergenic regions, up to 350 total base pairs. And of course, if there was any overlaps, we trimmed some of those overlaps, and then any gaps between different portions of the fragmented genes, we added ends to help fill those spaces. So at the end of the reconstruction phase, we had essentially an isolate with its individual intergenic and gene sequences. We then organized this so that way we had the before and the after intergenic regions flanking the respective gene. And the reason why we did this is we were hoping that once we used our whole sequence tokenization method, we could be providing some additional spatial context to the model. And hopefully the model would learn any relationships between the intergenic regions and the genes that may confer resistance to a given antibiotic. And you can see in the rightmost portion, that's essentially what we had going into the model was tokenized whole sequences. So an isolate-based sequence of tokens. After this, we went ahead and we went into the development of our model using our sequences. So we selected a BERT model, and the model that we used was based on the base BERT architecture. So we felt that because it's a classification task, felt like a decent model to use for this task. So if you're not familiar with how the BERT architecture works, I'll just briefly go through it from a high level. A given tokenized sequence is fed to an embedding layer. We use a dimension of 768. The positional encodings are then concatenated to the tokenized embedding or the token embedding to create a concatenated embedding that we can then feed to the next portion, which is the 12 transformer layers, each with multi-head attention and feed-forward network of dimension 3072. The output of this is a contextualized embedding that we then feed to the linear layer, which outputs a output logit where we applied a sigmoid function and then thresholding to obtain a prediction on if a given sample was resistant or susceptible to a given antibiotic. Additionally, we can extract from the multi-head attention the attention scores, and we can, it's discussed more in detail in the paper, but we can perform downstream token importance analysis to see if there's any relationships between important tokens and possible genes of interest. So after this, I'll just introduce some of the parameters we use to train. So we use a batch size of 256. The maximum number of epochs that any given model could be trained was 200. And we use cosine annealing to vary the learning rate over the course of training, hopefully leveraging the more exploratory high or larger learning rates and the more refining nature of the smaller learning rates. For regularization, we use the dropout of 0.20 and a weight decay of 1e to the negative 4. We trained each model for each antibiotic for 5 folds of the development set. However, we also wanted to look and see if the intergenic regions also were important when it came to the model's performance. So we also looked to see through cross-validation if that was of value. And then the final step after developing our model is, of course, evaluating the performance of the model on the evaluation set. So we can apply our model to the evaluation set, and that gives us predictions, but it doesn't really tell us how well our model's performing in relationship to other established tools in this space. So we selected 5 other tools to compare to our model in terms of predictive performance. And they are TB Profiler, Microbe, ResFinder, MTB++, and KBAR-Q. And it's worth noting that MTB++ was also trained on the same training data that we use to train our model. It's also the only other method here that is machine learning based, so it uses both a logistic regression and a random forest alongside K-mer and feature analysis to provide an output. So we felt like this was a fairly decent or a good baseline to compare our model to in terms of performance on this holdout set. And then from the prediction, F1 scores to compare our model to these other tools. And this is mainly because the other tools are a bit more deterministic. They use things like curated lists of mutations or data stores or databases to help provide an output on resistance for MTB. So I mentioned previously that we looked to see how the model would perform with intergenic regions included or excluded. And so this is over 5 folds of the this is the average performance over 5 folds of the training data for each antibiotic, with the excluded intergenic regions in blue and included in red. And what we saw was that including the intergenic regions didn't really impact performance too much on the first line antibiotics and for some of the second line antibiotics. But for 3 of the second line antibiotics, namely levofloxacin, moxifloxacin, and ethionamide, we see that including them actually helped boost performance quite significantly. We also saw that including the intergenic regions for linezolid, one of the last resort antibiotics, seemed to help. But the other last resort antibiotics don't really see too much of a difference. Actually, dolaminid even sees a little bit of a performance decrease. But given this result, it was a good indicator that we should include the intergenic regions when training and developing our model as well as testing them. So after this, we had our models and we decided it was time to apply them to the evaluation set and just see how they performed in comparison to the other methods that we discussed. So on the left-hand side, in the most left-hand, I guess, column there, you'll see the performance of our method on the evaluation set. And for the most part, LLMTB performs quite comparatively to the other methods included in this study on the first and second line antibiotics. Now the data imbalance is the antibiotics in the first line the first line antibiotics, the data sets are much more balanced. So we see that for rifampicin and ionizid, those 2 top rows, we see that the model performs quite well. But then on the second line antibiotics, there is a bit of a decrease in performance, and there's a larger spread, and that's likely due to a larger imbalance in the data set. But we still see that these are pretty competitive results to some of the other methods. For the last resort antibiotics, that's where we see the substantial decrease in performance, and this is seen across all of the all the other methods included in this study. And this is likely due to these antibiotics, resistance to them is quite rare, so their imbalance in the data set is quite large. So some of these only have like a handful of samples, a couple hundred samples out of thousands. So that's likely why we're seeing this decrease in performance with the large language model. However, comparing LLMTB to the other machine learning-based method in the study, and just to recap, it's also trained on the same training data. So neither one of these models has seen this holdout data. We see that LLMTB performs better than MTB++ on 10 of the antibiotics, and that's to a varying degree. Some of them are closer in performance numbers than others, but notably, LLMTB performs quite substantially better than MTB++ on the last resort antibiotics. And on 2 of the last resort antibiotics, MTB++ was able to acquire no correct resistant predictions. So this was a good indicator that the LLM is likely learning or using some more nuanced information in these sequences to provide an output. And before I just go over some of the performance numbers between our method and the other methods included, I'll just mention that the other methods don't always provide an output on the set. Now, because these are more deterministic, there's a bit more inflexibility. You could apply, say, LLMTB to one of these other antibiotics that's not included and probably get an output. It might not be correct, or the performance might not, we haven't looked at the performance in that regard, but it's just to note that these other tools don't always provide an output. It's to a varying degree. So TB Profiler seems to be one of the more comprehensive ones, providing an output on 12 of the 13 antibiotics, while KBAR-Q only provides an output on 5 of the 13 antibiotics. And in the rightmost column, you'll see the delta, or the difference between our method and the best performing method for any given antibiotic. So in the two columns where you see 0, that's where LLMTB is actually the best performing method. And that was on rifabutin and chlorphazamine. So, but we also see that for the first and second line antibiotics, LLMTB is within 3 points of the best performing method for any given antibiotic. And in some cases, it's less than a point difference to the F1 scores. So quite comparative to the other methods in the first and second line antibiotics. However, for the last resort antibiotics, again, we do see a much larger difference between our method and the best performing method, which more often than not was TB Profiler. But it's not performing the worst in all cases. So we do believe that by acquiring more data for these antibiotics, we may be able to boost performance here. And it's definitely an avenue that we're exploring going forward and trying to see if we can squeeze more performance out of those last resort antibiotics. But yeah, so this kind of wraps up the discussion on our model or our method. And yeah, I open the floor to questions and hopefully I can answer them.
Yeah, thank you very much for this really nice presentation. Here's already a question.
Thank you very much for your presentation. I actually have two questions. So the question number one, when you do check the performance, the sample's resistance, is it like really the strain of this resistance to antibiotic, or just an empiric observation like that person who had that strain was resistant or not?
So the data set itself is so what they did was that most of the labs that provided data to the cryptic, the cryptic data set, they used, there were 3 reviewers, and one of them was, I believe it was a Thermo Fisher tool. So in some cases it was somebody actually doing the AST and recording okay, this is a resistant, and that's usually based upon some guideline or a threshold value that's been predefined.
Yeah, I asked this question because if it's an empiric observation, they just notice that the person with the tuberculosis took that antibiotic and it didn't help. Mm-hmm. The resistance could have been not in the Mycobacterium tuberculosis, but for example, if the antibiotic is administered orally, the resistance could have been in the gut mm-hmm. microbiota. In that case, maybe you can repeat that, but with a DNA from gut microbiota and find some resistance. And, okay, I will ask you more questions in the break. Thank you.
Hello, and thank you for your interesting work. And thank you for providing all the baselines. That was very rigorous and very good to see. So if I was a clinician, which I actually am, I would trust TB Profiler because they have better performance, right?
Yeah.
So can you maybe put a bit your work into context with that? So how do you see the importance of your work? And maybe is there any possibility how your technique can leverage maybe TB Profiler, or they can get the best together out of that?
So this is something that we've actually discussed, and we're curious to see if we can somehow look and see how TB Profiler is providing those, its performance. It's using a database, so those resistance mechanisms are documented somewhere. So if there's a way that we can maybe guide the model to focus on those, it might be helpful. It might help with performance improvement. So that's something that we've discussed, and we're definitely looking as future work. Yeah.
Yeah, that would be great. Mm-hmm. That could maybe improve the overall function.
Mm-hmm. Yeah.
Thank you for the nice talk. I had a question, as you mentioned that you can still evaluate the features. So did you look into the important features that are necessary for prediction?
We did. I have a separate slide for this as well. I didn't know if we'd have time to get to it. But so any of these genes or intergenic regions that you see that are underlined and bold are genes of importance to the model when predicting, that were also, I guess, they're supported by literature. So it is using genes in intergenic regions that are known to confer resistance.
Then isn't it like, because there are specific mutations in these genes which are responsible, but can it also pinpoint it somehow? Because yeah, you can get the gene, but not the exact mutations, right?
Yeah, so because we've done we're doing the whole gene tokenization, I can get the gene, but I can't tell you where specifically in the gene. So that's another thing that it's a limitation of this method, and we've looked into other tokenization techniques, and it's something that we're currently working on right now, as a way of possibly trying to do that, because as you mentioned, I can't tell you where, so.
Okay, well, I just have to say, I really enjoy your talk. Also, the performance estimates that you highlighted where other methods are strong. So you have this really comprehensive picture. It's really interesting. So thank you for that. Unfortunately, no more time for questions, but I'm sure he's available in the break. So let's thank Conrad again.
Thank you.